This visualization tracks the staggering toll of a disease we have been fighting for millennia. In 2023 alone, there were an estimated 10.8 million new cases of tuberculosis globally, resulting in 1.25 million deaths. It remains the leading single-agent infectious cause of death in the world. Since 1921, our primary defense has been a single tool, the Bacille-Calmette-Guerin, or BCG, vaccine. Over the last century, it has been administered more than 4 billion times. But BCG's protection profile presents a stark paradox. When given to neonates, it reduces severe pediatric forms of the disease, like tuberculosis meningitis, by up to 80%. However, its efficacy against adolescent and adult pulmonary TB is highly variable, often dropping to near zero. This failure in adults is the critical missing piece in eradication efforts. As you can see in this chest X-ray, adult pulmonary TB causes extensive bilateral cavitary lesions in the lungs. Those specific lesions are the principal drivers of global transmission. This chart shows the modern clinical pipeline, which is finally moving past the century-old benchmark. We now have a biologically diverse set of vaccine candidates advancing through clinical evaluation, spanning from live attenuated strains to the first mRNA platforms. To halt the spread of the disease, we can no longer rely on simply distributing more BCG. Eradication requires shifting our vaccine targets, moving from simple blood-based T-cell counts to the complex architecture of lung-specific immunity. To understand how the field is engineering that new immune response, we have to look at the most informative clinical failure of the modern era, the 2013 MVA-85A infant trial. The scientific premise of this viral vectored vaccine was straightforward. MVA-85A was designed to induce massive CD4 plus Th1 cell responses and high production of the cytokine interferon gamma, and it did exactly that. Despite generating robust antigen-specific Th1 cytokines and high interferon gamma levels, the vaccine failed to protect infants against infection, yielding an efficacy of just 17.3%. Vaccine development usually relies on a correlate of protection, a measurable immune marker, like a specific antibody titer in the blood, that reliably predicts whether the vaccine will succeed in the real world. The legacy of the MVA-85A trial is that it proved antigen-specific Th1 cytokine secretion is not a sufficient correlate of protection for TB. This result demonstrated that the field's existing model for predicting vaccine success was incomplete. High T-cell counts in the blood simply did not translate to immunity in the respiratory tract. To build the modern pipeline, immunologists realized they had to move beyond the narrow Th1 paradigm and map exactly how the bacteria weaponize our early innate defenses. The infectious dose of Mycobacterium tuberculosis is incredibly small. A human only needs to inhale roughly 3 microscopic bacilli to trigger a cascading systemic infection. Looking at this diagram of the natural history of infection, we see an immediate paradox. Inhaled bacilli are taken up by frontline alveolar macrophages, but instead of destroying the threat, the bacteria actively arrest phagolysosomal maturation, neutralizing the cell's internal acid attacks. To survive the toxic chemical environment inside the macrophage, the bacteria adopt a specialized antioxidant transcriptional signature, effectively detoxifying the host's reactive oxygen species. Next, the bacteria impair dendritic cell maturation and cleave immunogenic chaperones. This strategically delays the priming of our adaptive T cells by up to 8 weeks. Their most insidious tactic, inducing cell death in bone marrow myeloid progenitors, actively erasing the innate immune memory that earlier exposures might have installed. This sequence explains why standard vaccines fail. They rely on an adaptive immune system that MTB actively blinds, delays, and turns into a protective fortress. This diagram illustrates the multi-armed defense model scientists are constructing. Layer 1 is trained innate immunity, reprogrammed alveolar macrophages that restrict the bacteria. Layer 2 relies on lung-specific tissue-resident memory T cells. Using aerosol delivery, vaccines can generate high frequencies of these cells to colonize the airway and intercept bacteria at the site of infection. Layer 3 involves Th17 cells. These are crucial for early control because they recruit neutrophils and organize the defense architecture. However, this layer requires precise regulation. Sustained, overactivated Th17 responses drive severe neutrophil-mediated tissue damage. Layer 4 rehabilitates humoral immunity. We now know that heavily exposed individuals who never show infection possess distinctive class-switched IgG and IgM antibodies. These Fc functional antibodies physically tag the bacilli, shaping intracellular trafficking and heavily enhancing the macrophage's ability to kill the bacteria. Achieving true sterilizing immunity against TB requires the precise orchestration of all 4 layers, moving vaccinology far past simple T-cell activation. If we zoom out to the tissue level, shown in this cross-section, we see the macrostructure where this battle is ultimately decided, the granuloma. Granulomas are not uniform containment walls. They are highly dynamic, lesion-specific microenvironments that act almost independently of one another. A remarkable paradox. Within a single host, some individual granulomas can successfully sterilize the infection, while neighboring ones simultaneously fail, progressing to active cavitary disease. Because clinical outcomes are determined locally at the lesion scale, immune measurements taken systemically from the blood consistently underpredict actual protection. This granuloma paradox is the primary reason the field still lacks a validated correlative protection. As outlined in this trial endpoint table, measuring vaccine efficacy currently relies on 2 primary strategies: prevention of infection, or POI, and prevention of disease, POD. Historically, POI trials were preferred because infection events are about 10 times more frequent than clinical disease, allowing for smaller, shorter trials. But after a string of recent high-profile failures, these surrogate endpoints have severely underdelivered. Consequently, the field has universally converged on prevention of disease endpoints. These measure bacteriologically confirmed pulmonary TB, which is exactly the metric policymakers care about. But without a validated immune marker to act as a shortcut, running POD trials means relying on massive, multi-year, multi-country studies. Development remains strictly empirical, heavily extending both the timeline and the cost. Despite those methodological hurdles, a vanguard of late-stage candidates currently anchors the mid-2026 pipeline. This profile details M72/AS01E-4, a protein adjuvant subunit vaccine currently fully enrolled in a 20,000-participant Phase 3 trial. In its Phase 2b trial, it demonstrated an efficacy signal of roughly 50% protection against active pulmonary TB over 3 years in infected adults. This section outlines VPM-1002, the most advanced recombinant BCG candidate. It is engineered to delete urease C and express listeriolicin O, allowing the antigens to escape the phagolysosome and improve MHC1 cross-presentation. As recorded in this timeline of trial results, VPM-1002 recently reported its PREVEN-TB Phase 3 readout. While it missed statistical significance against all TB forms, it showed a 50.4% efficacy against extrapulmonary TB, with even stronger signals in younger demographics. Finally, this table tracks MTBVAC. Unlike BCG, which is derived from cows, MTBVAC is a live attenuated human MTB strain. Crucially, it retains the full ESAT6 and CFP10 antigen repertoire that was lost in BCG over a century ago. The biological hurdles of tuberculosis are immense, but by systematically layering trained innate immunity, tissue-resident memory, and precise antigen selection, these engineered platforms are finally moving the field past the 100-year BCG benchmark.